Welcome back to the series. In episode 15, we're stepping inside the test tube to explore how a tiny piece of a plant can grow into an entirely new plant. We're talking about totipotency and the basics of plant tissue culture, one of the most fascinating concepts in plant biotechnology. So, let's get started. You can take one leaf cell from a tomato plant and grow an entire new tomato plant from it, not a clone of the leaf, but a complete, fruit-bearing, fully functional plant. That is totipotency, and it is one of the most remarkable properties in all of biology. Today, the science and sweeping applications of plant tissue culture. Totipotency, the property animals lost. Every plant cell carries the complete genome of the entire organism. Under the right conditions, any living plant cell can unwind its developmental restrictions and regenerate into a full plant. This is totipotency, the total potential to become anything. Animals largely lost this property after embryonic development. Plants kept it forever, and we have learned to exploit it to feed, clothe, and heal the world. How big is this industry? Plant tissue culture is a global industry worth over $6 billion annually. It produces banana plants by the billions. The entire commercial banana industry depends on it, disease-free seed potatoes, orchids at industrial scale, pharmaceutical compounds from plant cells, and transgenic crops including much of the world's soy, corn, and cotton. This technology is quietly everywhere. Here's a quick question. Why can a single plant cell regenerate into a complete plant, while most animal cells cannot? Is it because animal cells lose their genetic material? No, animal cells generally retain their complete genome, but during development, many genes needed for full reprogramming become epigenetically silenced. Plants, however, retain much greater developmental plasticity, allowing certain cells to reactivate these developmental programs under the right conditions. And that remarkable ability is what we call totipotency. From theoretical principle to global industry. Now, let's take a quick look at how plant tissue culture developed over the years. It began in 1902, when Gottlieb Haberlandt proposed the idea of totipotency, that a single plant cell could potentially regenerate into a whole plant. In 1934, Philip White successfully cultured tomato root tips, showing that plant tissues could be maintained and grown in culture. Then, in 1958, Frederick Stewart achieved something remarkable. He regenerated complete carrot plants from single cells, providing strong evidence for totipotency. In 1962, Murashige and Skoog developed the famous MS medium, which is still widely used for plant tissue culture today. Later, in 1983, agrobacterium-mediated transformation helped establish the foundation for genetically modified crops. And by 1994, the flavor-saver tomato became the first commercially marketed genetically engineered food crop in the United States. Today, tissue culture is used on a massive scale, from producing disease-free plants to rapidly multiplying crops like Cavendish bananas. What started as a scientific idea over a century ago has become an essential tool in modern plant biotechnology. Now, let's look at the 5 common types of plant tissue culture. First, meristem culture, using shoot or root tips to produce healthy, often virus-free plants. Next, callus culture, where plant tissue forms an undifferentiated mass of cells that can later regenerate into new plants. Then we have protoplast culture, which uses plant cells with their cell walls removed. This is useful for cell fusion and creating somatic hybrids. Embryo culture starts with immature embryos and can help rescue hybrids that might otherwise fail to develop. And finally, anther culture, which uses pollen-containing anthers to produce haploid plants and speed up the development of pure breeding lines. So, the starting material may differ, but the goal is the same, using plant cells and tissues to create new possibilities in plant biotechnology. So, where is plant tissue culture actually used? The answer is, almost everywhere in plant biotechnology. In agriculture, it helps produce disease-free plants and rapidly multiply valuable varieties, on a scale of billions of plants each year. In horticulture, it's widely used for crops like orchids, strawberries, and bananas. In pharmaceuticals, plant cells can be grown in bioreactors to produce valuable compounds such as Taxol, artemisinin, and chiconin. It also plays an important role in GMO development, where tissue culture can help regenerate plants after genetic transformation. For conservation, tissue culture provides a way to preserve the genetic material of endangered plant species. And finally, in research, it helps scientists study genes, transform model plants like Arabidopsis, and understand how plants develop. So, from farms and laboratories to pharmaceutical production and conservation, plant tissue culture has become a remarkably versatile technology. Here's a quick concept check. Why does the commercial banana industry depend so heavily on tissue culture instead of growing bananas from seeds? Is it because banana seeds are too expensive? No, the popular Cavendish banana is a sterile triploid, so it doesn't produce viable seeds. Instead, bananas are propagated vegetatively, and tissue culture allows growers to produce huge numbers of uniform, disease-free plants quickly. But there's a catch. Because these plants are genetically very similar, a disease that can overcome their defenses could potentially threaten banana production on a massive scale. From leaf to lab, the complete Explant workflow. The tissue culture process begins with selecting and sterilizing an explant, the initial plant material. Contamination by surface microorganisms, fungi, bacteria, endophytes, is the primary challenge in plant tissue culture. The sterilization protocol must kill surface organisms without killing the plant cells. Surface Sterilization Protocol 1. Select explant, young, healthy leaf tissue, node, or shoot tip from actively growing plant. Avoid old or senescent tissue. 2. Wash under running tap water for 10 to 15 minutes to remove gross surface contamination. 3. Ethanol dip. 70% ethanol for 30 to 60 seconds. Kills most surface microorganisms. Brief to avoid tissue damage. 4. Bleach treatment. 10 to 20% sodium hypochlorite solution for 10 to 20 minutes with gentle agitation. The most critical decontamination step. 5. Triple rinse with sterile distilled water, 3 times 5 minutes. Remove all bleach. Residual bleach kills plant cells. 6. Transfer to laminar flow hood. Trim edges of explant with sterile scalpel to expose fresh unsterilized tissue. Place on agar medium. So, what actually happens after the explant is placed into the culture medium? During the first few days, the explant settles in and may darken slightly as it responds to the wound. By week 1 to 2, the cut edges begin to swell as cells start dividing and taking up hormones from the medium. Around weeks 2 to 4, callus tissue begins to form as cells return to a more flexible, meristem-like state. By weeks 4 to 6, shoots or roots can begin to develop, depending on the hormone balance. Between weeks 6 and 10, plantlets start forming with organized shoots and roots. Finally, around weeks 10 to 14, the plantlets are transferred to rooting conditions and gradually acclimatized to the outside environment. So, what starts as a tiny piece of plant tissue can, over several weeks, become a complete new plant. The hardening off problem. Plants grown in tissue culture live in a perfect, sterile, humidity-controlled environment. When transferred to soil, they face non-sterile conditions, lower humidity, fluctuating temperature, and physical stress. This transition, hardening off, is often the biggest bottleneck in commercial micropropagation. Up to 30% of plants can be lost at this stage if the transition is not managed carefully. 3 mistakes in the banana industry. Mistake 1. Under-sterilizing the explant. The fix. Surface contamination is the #1 failure mode in plant tissue culture. If contamination rate is above 20 to 30%, increase bleach concentration, extend contact time, or add a surfactant, 0.1% tween 20, to improve surface penetration. Keep a contamination log. Track which plant species and tissue types are most problematic. Mistake 2. Wrong hormone balance for the desired output. The fix. High auxin gives callus and roots. High cytokinin gives shoots. Equal amounts give indifferentiated callus. If you want shoots and get callus, your cytokinin-auxin ratio is wrong. Always consult the specific protocol for your plant species. Hormone optima vary enormously between species and even cultivars. Mistake 3. Endophytic bacteria contamination. The fix. Endophytic bacteria live inside plant tissue and cannot be removed by surface sterilization. They typically appear as bacterial colonies on the medium surface 1 to 2 weeks after culture initiation, looking like slimy translucent growth around the explant. Add broad-spectrum antibiotics, cefotaxime, timentin, to the medium for the first 2 to 3 subcultures to suppress endophytes while the clean culture establishes. The Cavendish banana story is a lesson in monoculture risk. Because every commercial banana is a genetic clone, the entire global supply is vulnerable to any pathogen that can infect Cavendish tissue. Panama disease, Fusarium wilt, TR4 strain, is currently spreading through banana plantations globally. Plant tissue culture researchers are working on TR4-resistant transgenic or hybrid banana varieties, which will themselves be produced and distributed using tissue culture micropropagation. It's time for the final quiz. A plant biotechnology company wants to produce disease-free strawberry plants at scale. So, which tissue culture pipeline would you choose? Start with meristem tips from selected plants, sterilize them, and establish them in culture medium. Then multiply the shoots, root them using an appropriate low-oxen medium, and gradually harden off the plantlets before transferring them to the commercial nursery. This approach allows one elite mother plant to produce thousands of genetically uniform, disease-free plants. And that's the power of plant tissue culture. Summary. 1. Totipotency means every plant cell has the potential to become a full plant. Unlike animal cells, plant cells retain the epigenetic flexibility to reactivate developmental programs under the right hormone conditions. This is the biological foundation of all plant tissue culture. Understanding it explains why hormone ratios, explant choice, and medium composition all matter so profoundly. 2. Plant tissue culture produces crops, drugs, and conserves biodiversity. Bananas, orchids, strawberries, potatoes, industrial scale production. Taxol, artemisinin, pharmaceutical production from plant cells. Endangered species, preserved in germplasm banks. The range of applications from one basic technology is extraordinary. It is one of the most economically impactful biotechnologies that most people have never heard of. 3. Surface sterilization is the critical technical hurdle. Master it first. The most common failure in plant tissue culture is contamination from inadequate surface sterilization or endophytic bacteria. Perfect your sterilization protocol before optimizing hormones, medium, or regeneration conditions. A contaminated culture cannot be saved by the best hormone ratio in the world. And that brings us to the end of episode 15 of our cell culture series. Thank you for watching. In the next episode, we'll dive into MS medium and plant hormones, and understand how nutrients and hormones control plant growth in culture. See you in the next episode. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.